So students, we have with us, sorry for this, Dr. Ranjeet Kumar, who is, presently working as a senior research engineer at Chalmers University of Technology, Gothenburg, Sweden. And he's passionate about societal change that can be brought about by scientific thinking, entrepreneurial pursuits, and environmental sustainability. Ranjit would like to contribute to the interface between youth science, society, environment with an objective to build a better world. And he is actually focused on understanding the molecular events involved in protein folding, misfolding, and disease manifestation. And he is also interested in metal and small molecular interaction with proteins and their functional re, uh, reproduction, both in eukaryotes and prokaryotes. And recently, he is looking for natural compounds, small molecules, and interacting protein that can influence amyloidogenesis. So, sir, pardon if there is many things to include in your profile, but
Yes,
whatever profile I have received from professor, I am just the
yes, I will give a short introduction
yeah,
of
yeah.
my journey into science.
Yeah, yeah. This is
So,
very
so
brief introduction,
yes,
guys. Very warm welcome, sir. On behalf of Srishti, delighted to have you with us. Over to
thank
you.
you, thank you. So I will just start sharing my slide.
Okay.
Can you see the slide now?
Yes, sir.
Yes, sir.
So, as thank you, Mega, for the nice introduction. And uh, my name is Ranjit, and I come from Jharkhand, actually, in very close to the capital city, Ranchi, in a place called Hazaribagh. And today, I would like to thank my parent university, Chalmers, and also Honeybee Network for giving me an opportunity to share my knowledge in the area of proteins. And the topic for today is protein factories, expression, purification, and application. And I extend my warm welcome to all the participants at the Biotech Innovation Ignition School. So, before I start my talk, I would like to give a small introduction of how I navigated in the field of science. So, I would like to first bring into context my parents, my mother and father, who had a strong faith in me in whatever discipline I choose. So, I started my formal education actually started in class 7th. Before that, I was like my father was in a transferable job, and every 6 to 8 months he used to get transfer. So I didn't get an opportunity to continue education as I was supposed to do. But then after he decided that we will be settling in a town called Hazaribagh because it has good schools and colleges. And there I started my education from St. Xavier's school. After my schooling, I went into a college, St. Columbus College, which at that time, we like maybe almost 20 years back, they started a program on biotechnology, and biotechnology at that time was fairly a new subject with no defined career path at that point of time, but it was like a fascination term for many. So I wrote an entrance and I entered and into my bachelor's program at there, and it was a part of the Vinova Bhave University. After I completed my bachelor's, I had I was looking for opportunities, uh, and with a biotechnology degree, it seems that the only way to move ahead is to get a master's degree. So I ended up going to University of Calicut, and in Kerala, I was very homesick. Homesick means that I haven't moved off Hazaribagh before that. So it was my first opportunity right from the north of India, moving to the extreme south of India, and also a cultural shock. So I went there, and of course, at that time, the food pattern and everything was so different in that part of the country that led me to a lot of challenges. But I eventually passed from University of Calicut, and then the way ahead was that when you do your masters, the only way ahead is at that time was that, okay, if you have done your masters, now what lies ahead? Because there are very few biotech companies in India at that time. The idea was to go for higher studies, and the higher studies would mean that you will end up doing a PhD. So I cleared um, all the entrance examination that was required at that time, and one of them is Council for Scientific and Industrial Research Fellowship that I cleared. And I also got decent training during my masters, where it was like kind of a first exposure to the field of biotechnology. I got training, one as a part of summer training during my masters in IMTech Chandigarh, which is Institute of Microbial Technology, and the project in my masters was done at Institute of Life Sciences Bhubaneswar. So it was like opening my eye into the realm of research. Then I, for a small period of time, I went to as a project assistant in NIRRH Mumbai, which is a National Institute for Research in Reproductive Health. It's a ICMR funded organization. And then I moved to CDRI, Central Drug Research Institute in Lucknow, under the mentorship of Dr. Vinod Bhakuni. He is a he was a Bhatnagar awardee, and he is no more. He died very young, and I was the last student perhaps to be awarded PhD under him. And there I worked on the area of protein folding and misfolding and understanding proteins. So after my and in CDR, I also get an opportunity to train myself in 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 a very innovative program which is called Technology Led Entrepreneurship Program, run by IIM Bangalore. And it was like an eye opening, because then at that time we got uh, an idea of how would you apply the knowledge that you have in science for the betterment of the society. So you should not be confined only to thinking about what happens inside the lab. The science should also have a societal touch, and that was the thing that happened. And eventually, like once I got my PhD degree, I was looking for an academic job, like looking for a position within India, and I had no plans to move abroad. But as destiny would take me out, I ended up completing my PhD from CDRI, and the degree was awarded by JNU New Delhi. And I, like when I went to my boss and asked that I want to work in India, he said that first you should have some working knowledge in Europe. He didn't say specifically US. He said Europe. And I realize why he says so now. And I can tell you at the end of my talk what is the difference. So I ended up coming to one of the oldest university in Scandinavia, Uppsala University. And it was like a big leap for me. A guy who is more or less homesick, who was reluctant to move to Kerala also, has to move abroad. And there was lot of adjustment issue when I joined here. Leave apart the science, but other thing, climate and all those things were making me like I wanted to revert back. And in that case, I was underperforming, I thought, and I did some excellent work there. It was more of a learning out there, uh, and then I wanted to move back to India, but then uh, I got again an opportunity to work in a Swedish university, and since my first experience was not good, and this was something which was very close to me, because it was a project which was related to environmental science and biotechnology, and I took this opportunity in Örebro University, which is a sister concern of Uppsala University, and I worked there for 2 years, ready to return now to India. But again, at that time when I planned to return to India, because you cannot make a short term plan when you are abroad. So I was looking through different opportunity. And since I disgraced a little bit from my field while joining Oribro University, because I was trained as a protein chemist, and I moved into molecular biology, hardcore molecular biology, when I did work in Oribro University. So I wanted to have a little bit more on protein side, and I ended up going to uh, uh, Russia actually. And this happened in a uh, very funny way, because uh, the professor was from Switzerland, and we thought that we will be landing in Switzerland, but he was establishing a uh, facility in Russia, and the project was called nanopumps. I went there and learned a lot. It lasted for a year, because then the professor himself thought that it was not a good idea to start something in Siberia actually, not even Russia. It's in Siberia, in a city called Tomsk. So to cut the long story short, I again went to my parent university as a faculty, as a guest faculty, and taught there for 6 months. But the bug for research was already within me, and that brought me back to Sweden again. So I came to Chalmers um, as an expert in protein chemistry, and I started here as a research engineer in the team of a researcher, Pranila Vitung Staffshed. And I came here in 2016, and I am continuing here uh, as a as a senior research engineer at present. And this gives me an opportunity to learn more, explore more universities in Sweden, and my journey of learning continues from here. But this is just a slight introduction, and now I will move into the topic. If I am very fast, just let me know. And also, I would like you to interrupt in between if you don't understand anything. So here I am. I have already introduced myself, so I don't like to tell much. But presently, I am researching on protein folding disorders, mostly amyloidogenesis, especially in the area of Parkinson. In Parkinson, what happens that there are some plaques, protein deposit within our uh, our head, and these protein deposit leads to loss in memory and motor related function, that the movement of your body will be affected. And we are studying and trying to study the key culprit, which is a protein, and trying to find out ways and remedies to uh, uh, actually slow down this process. And this we do mostly in vitro studies on purified proteins. So when we want to talk about protein and purifying protein, I just want to bring into the context, how does the picture inside the cell look? So if you look into the cell, it will look like something like this. So I took it from Trends in Biochemical Sciences, this picture, this and here you see that there are a lot of molecules that is crowding within the cell, and it's a picture of a bacterial cell. So to oversimplify it, if you see that you want to work with a small protein, but inside the cell, apart from the protein, there are a lot of other things sitting. There are carbohydrate, other macromolecules, nucleic acids, ribosomes, and everything is going simultaneously within the cell in a very crowded environment. And your approach is to get a purified protein. And why you want to have a purified protein, I will come later. So if you want to study protein, uh, uh, they are chains of amino acids. And these amino acids, when you see it as a chain of amino acids, this is called the primary structure. The primary structure of the protein undergoes uh, undergoes some bonding, and it forms the secondary structure. And the secondary structure is either alpha helix or beta plated seed. Some protein does not have any structure, and these protein are called intrinsically disordered protein. And then comes the tertiary structure. Tertiary structure is a fully folded protein, and where alpha helix and beta sheet part of the protein uh, form a fold, and this fold can either be like it the protein can either be globular, like a ball, a small folded structure, or it can be fibrillar. Fibrillar would mean like there are proteins like collagen, actin, myosin. These are filamentous proteins, so it can also be like that. And finally, these tertiary structure can can assemble together to form a quaternary structure. And one good example is that you have these globine molecules, uh, and four of these globine molecule interact together to with a heme moiety, a heme moiety to form hemoglobin. So hemoglobin is made of four different units of protein. So that gives you the quaternary structure. So if you go from primary to secondary to tertiary and quaternary structure, one thing to remember here is that when you see primary structure, it's like in a local parlance, if we talk, it is like alphabets. But when it assumes its secondary structure, it's like a word. When it moves from secondary to tertiary structure, it's like a sentence. Uh, and eventually it forms paragraph when two or three molecules come together and interact with each other to form a macromolecular structure. Yes, and then I want to show you one thing which we work here mostly. These are protein. This is a AFM image taken by me, and these are proteins. It's a atomic force microscopy whereby you you put the protein on a mica chip and you try to scan it through the microscope to see how does it look. And these are long stretch of a protein which is alpha synuclein, which is a key culprit uh, in Parkinson disease. So if you go for the structure, it's like this. It has some uh, alpha helical structure, and this alpha helical structure comes into play when it interacts with a membrane counterpart in the cell. And eventually what happened that there are small stretches of repeats within this 140 amino acid protein, which stack one above the other like a stack of books, and due to their beta seats in the protein, they form long chains like structures in the form of fibrils. So this is the area where we are researching at present moment. We are studying this particular intrinsically disordered protein. So why purify protein? Why do we want to purify protein? Why there is a need to purify protein? So first is that within the cell they act as enzyme and they catalyze many of the metabolic reactions. Like if you read Krebs cycle or any other metabolic cycle, you will come across several enzymes. And if you want to study those enzymes, it is important to purify them and work with them. Then there are certain structural elements. A structural element like basically actin, collagen, myosin, and these structural elements should be studied in detail to know more about their function and how do they behave. So you need to purify the protein and work with them in vitro. Then there are receptors, and these receptors uh, uh, are there on the surface of the cell and they interact with the protein or other molecule. And this receptor ligand interaction, if you want to study and if you want to see, or if you want to silence some receptor or which can interact with something unwanted, which can cause a disease or infection, then it's important to study this receptor. Also, within the cell, there are intracellular signaling that goes on. So there are many protein involved, and it works like a relay race. So proteins are like baton, and one baton passes hands to the other runner, and it regulates many of the biochemical processes within the cell. And I am sure most of this topic would have been dealt in this program also. And then they are key component of many of the machineries within the cells. They keep the cell intact, and uh, they uh, if you study protein synthesis, there are a lot of proteins which act in transcription, translation, and post translational modification of the protein. So oh, when you want mm, with the advent of new technology, one can work with DNA and RNA, and this has been made possible. And the era of in the era of recombinant DNA technology, one can make any protein, any desired protein, and this can be done through several enzymes, and these enzymes need to be made. So that's why it is very, very important that we should have the ability to isolate and study purified protein. And this is like lies in the heart of modern biochemistry. So one thing I also want to bring here is that it doesn't it's not only like a academic pursuit, but it's also an industrial pursuit, and I will tell you how. So this is a old data, but if you see the prices of the protein, purified protein, so bovine growth hormone per gram of the protein will cost this much, 14, and granulocyte colony stimulating factor cost this much. So the idea is that if you have purified protein, there are many uses to it. You can have it as a biochemical reagent. They you can have biological activity. You can sell it as, like, now you have protein powders to make muscles. So uh, there are various aspects to it. One is that the academic and research pursuit, where you want to know the biological activity, you want to enhance the biological activity of the protein. And the other stuff is like many of the kits that you see in the market, even for testing of the corona, let's say, or for any of these ELISA based kits, they need biochemical reagent, and these biochemical reagents are enzymes, of course. And then if you want to study the interactions between receptor and ligand, then it becomes very, very important. And in the area of drug discovery also, it plays a very, very important and crucial role. So we call it structure function folding studies of the protein. So it has given us a new area of protein engineering, and with techniques like mass spectroscopy and nuclear magnetic resonance, and deciphering the crystal structure of individual protein and studying this protein ligand interaction, it becomes quite obvious to that one needs to purify the protein and work with the protein. So there can be various system in which you can purify the protein. Like uh, one well established and standard system is E. coli. Then you can also do it in yeast. It can be done in baculovirus system, and it can also be done in a mammalian cell. But the thing is that you have to do a cost benefit analysis, and also what is the purpose of purifying the protein. Supposedly, if you want a protein to be produced in a bulk amount to study the structural features, then E. coli is the organism of choice, because you can grow it in a really very inexpensive media, and every means it divides quite quickly. You just do a overnight culture, and with the gene of interest inside the bacteria, and you can purify like milligrams of protein. But yes, there are limitations, because bacteria is a primitive organism, and it doesn't have the mechanism to mm, post translational modification is something that is very important to the protein which are there in humans. So you cannot there are ways that you can do some modification through E. coli, but you cannot achieve it fully. So then comes into the rescue the eukaryotic system, and yeast is the best example. And I must tell you at this point of time that in Chalmers there is a very good facility. Actually, there is Chalmers is basically a, a engineering institution, but biotechnology is also a discipline, and it's a fairly new discipline that has been added uh, added, but there is a very good facility of synthetic biology and protein engineering here at Chalmers. So these two systems are rapidly being used, E. coli and yeast, for purifying protein and engineering protein. Uh, yes, then comes the baculovirus system. It's a kind of a insect cell line, and you try to express your protein, and this gives you a kind of a little edge, because you can you can get some post translational modifications using this. Uh, and then comes finally the mammalian cells. So mammalian cells are very very like I would say expensive setup, and you need to maintain these cells nicely. Sometimes we do it in mammalian cells, but usually, like when you need to prepare vaccines and things that need to be used for human use, then you go for mammalian cells, basically therapeutic proteins. And the reason for doing that is that when you grow in E. coli, there will be some small toxins that are produced by bacteria, because if you want to if you want to engineer bacteria to produce something which is not natural to them, they will try to avoid that, and in the due course they produce, and it's a inherent nature that bacterial surfaces have, and within the bacteria there are exotoxins and endotoxins, and these toxins can be lethal. So one has to, even if one scales up the productions in bacteria, it's more a kind of you need to remove that before you try to use them in the eukaryotic system and therapeutic use. So I will go in a very uh, what you have studied in your plus two. I want to start from there. So you must be familiar with the Lac Operon, and the difference between the protein synthesis mechanism in prokaryote, which is bacteria, and eukaryote like humans, is that in prokaryotes, genes, that is the fundamental units, they are arranged one after another as a battery, and there is no uh, there they are all uh, um, uh, they are all arranged in a way that they code. There are no introns there, but in humans there are exons and introns. So this system proved to be a better system for engineering the thing and making the bacteria produce the protein. And the most used method is using the Lac Operon. So the Lac Operon is a operon system which help the bacteria to produce uh, three enzyme, which is beta galactosidase, permease, and transacetylase, on induction with lactose. So this system, and if the lactose is missing from the system, the lac I will the promoter the repressor will bind to the operator site, and it will not allow the translation of the protein. But once you have a inducer, here it's lactose. So when lactose goes and bind, it 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 uh, knocks off the repressor from the operator side, and the gene is ready for transcription. And once the gene is transcribed, it is then translated into these three enzymes: beta galactosidase, permease, and transacetylase, and then they are able to metabolize lactose. In the similar fashion, this system is used, and in the downstream, say LAC-Z, LAC-Y, and LAC-A, here you can put your gene of interest and force the bacteria to make your protein instead of making beta galactosidase, permease, and transacetylase. And this phenomena of uh, uh, making constructs where you use this idea of Lac Operon and uh, put your gene of interest downstream to these operons to in order to produce the protein is called recombinant DNA technology, whereby you just take a small part of the protein, that the gene of your interest, and then you can put it. And a simple scheme would be to just clone this your gene of interest in a plasmid, And this plasmid, I'm not going in details of the recombinant DNA technology, but you you make it such that you have your protein of interest, and you do it in a vector. A vector is like a vehicle, and you put it within the bacteria, and this process is done also chemically. So chemically you can grow a bacterial culture, and uh, you can make them competent. Competent means just they can take small pieces of DNA. So to make them competent, you treat them with a mild solution of calcium chloride, um, and then there are pores that will open, and it can easily take up your gene of interest, and you can grow, you can plate it on the, this process is called transformation, when the bacteria takes the DNA. And this you can do, uh, you can grow these bacteria in culture, and after culturing this bacteria under a pressure, and this pressure is like when you do this, you introduce an antibiotic marker. The idea of introducing an antibiotic marker here is that you will be it will be easy for you to select the bacteria which has your construct, because then um, in frame of your protein there lies also a protein which can uh, which can which can chop off the antibiotics, and when it chop off the antibiotics, it has a ability to grow on the plate. And then eventually you do purify protein by breaking open the cell, and uh, we do it manually in gravity flow. When I started my PhD, and in a simpler setup in CDRI Lucknow, so we used to have a small tag with our protein, and the rest of the work you can do it in a small column, and I will go through it in detail. How do we do it? It's called immobilized metal affinity chromatography, whereby you load your sample, and the sample has, you can think it like a hook. So uh, your protein of interest is special because it has that hook, but other protein doesn't have that hook. So it gets hooked to the column, which you can easily strip. When you put every all the protein into the column, what happens, whatever unnecessary proteins and other stuff and cell debris are there, they will pass through the column, and your protein of interest will bind to the matrix, which you can eventually remove and knock off from the matrix by changing either the buffer condition, or you can have a competitive binding with another molecule which can knock off your protein. So if I want to present you a small scheme of how do we do this, so we take the bacteria and we put the bacteria to grow in a shaker flask, uh, mm, usually typically 200 rpm per minute, and we grow it till the bacteria grow to certain extent, and we measure the optical density of the bacterial growth, and then we put an inducer, and that inducer, what it does is that it shifts the cellular machinery of the bacteria and compels it to make drive all its mechanism to make your protein of interest. So in this process, what happened that your protein of interest will be made in bulk inside the bacterial cell. So what do you do next? Next day you come and harvest your cell. After harvesting, you break open the cell, and this removes the cell debris. It's a very simple scheme. It removes the cell debris, and you have the supernatant, and the soup has your protein of interest, plus it also has other proteins and molecules uh, that are soluble, that doesn't move into the pallet. So you load it into the column, and as I told you that you can do it through several means, and one mean is that you attach a hook to your protein, and this hook is, in our case, is a small stretch of histidine, 6 histidine, a hexahistidine tag, which can be which is just used to um, separate your protein. And once you have separated your protein, this can either be on the N terminal part of the protein, or you can put it on the C terminal side, and then you load it onto the column, and then you have a chromatographic system. Now all the things I will show you next, that what we have in the lab, and then you get a single peak which you can monitor, because If the protein has, say, aromatic residues like tyrosine, tryptophan, and phenylalanine, they have uh, uh, they have a specific absorbance at 280 nanometer, 280 to 75 nanometer, and you can monitor that. And by monitoring that, you can hook your column to a system, and you can run it in an automated program, uh, uh, and then you get this single peak of protein with you to work with. This is how we purify the protein. And to check the protein, you collect your protein and you send the protein for mass analysis. And when the mass analysis is done, you know that you have the right protein in your hand, because it will tell you the exact mass of the protein. Also, you check it on a SDS page, which I will show you later. So these are the two systems that we have in our lab here in Sweden. We have a system from GE. Now it's called Sitvia, the company. And this system that you see is called Acta Explorer. So you can very well automate the entire process of protein purification, and there is a lot of advancement in this area. So once you have your protein of interest in hand, you can do the downstream processing. Like once you have the protein, you can do the entire process in an automated workflow. So we have the two base system. One is this system that comes from GE. It's called Acta Explorer, and the other system is from Biorad, which is called uh, NGC, New Generation Chromatography. So whenever you want to purify a protein, uh, one important thing is that there is a lot of message which is hidden in the sequence of the protein, and it's important that you you should decipher what is that information. And to do that, if you already know the sequence of the protein, one very uh, intuitive or very easy bioinformatic tool is XPASI, and the program is called Prot Param tool. So you can take your sequence and you can just put your sequence within this software, and it will give you all the parameters that the protein has. That it will give you the uh, for any purification to be successful, you must know what is the molecular weight of the protein, how many amino acids are there, what is the amino acid composition, uh, how hydrophobic or hydrophilic is the protein, how many tryptophan residues are there, and these all important, and what is the extinction coefficient of the protein. All these basic parameters you can know from here. So um, this forms the very fundamental. If you know the sequence of the protein, then it forms the very fundamental before you jump into the lab to find a strategy to purify your protein. Sometimes it's very very helpful. Especially I have seen that uh, it will it will it will help you design your purification workflow, that what kind of purification strategy you should take. So I'll stop here for a while and ask you if you have any question or anything till now before I move ahead.
So the strategies that you have told as of now, those are for the proteins that we know. In case
Yes,
in case we have some novel protein or we are trying to isolate some new protein,
yeah.
then what are the strategies that we can utilize?
Then you have to like one way is to go for the sequencing of the protein that you have in hand, and this has been made very simple. Like if you can purify, if you have a protein, if you know that how you come to know about a new protein, it might have some function or something. So one way is to isolate that protein, and even if you don't know the sequence, but you can see it on a gel that you have a band, right? And you just cut the band and you generate an antibody against uh, that protein,
Eliza,
and
sir.
then you can eventually get it sequenced, and you will get to know the sequence of the protein, and then you can, like, it's like backtracking the thing. Then you know that once you know the sequence, you can have a better workflow, and you can uh, you can make a strategy of purifying that protein.
So ELISA
So
will be worked in this case also.
yeah, ELISA, you can identify that protein through ELISA, say. And what is ELISA? ELISA is just antibody antigen reaction, right?
Yes, sir.
And you know the sequence of the antibody, so you can knock off the protein that binds to that antibody, and then you can get it sequenced.
Okay,
So
okay,
that
okay,
is
sir.
the first step. Yes, you can do it through ELISA. That's what I am telling you, that you need to have an antibody against it, or you need to have some hook that can take that protein out from somewhere, and then you can move.
Okay, sir. Okay. Thank you, sir.
Yes. Any more question? If not, then I can move ahead and we can have a question answer session at the end. The purpose of stopping here was that now I go deep into the purification steps. This was just a little bit of introduction. So how do you extract the protein? The way is that there are, if you are lucky, if you are working with a protein which is extracellular, say, so, or you can make or design construct whereby a protein is thrown out of the bacterial cell simply through a signal peptide that you attach to your protein. It's not very easy for all the protein, but you can do for some. In that case, you don't need to bother. You just you pallet the cell, collect the media, and you concentrate the solution, and you have the protein in hand, and you do the downstream processing. The other way is that if you are expressing the protein within the cell, and one thing I forgot to mention, that you can also, like, apart from bacteria, yeast, baculovirus, and mammalian cells, you can also express protein in plants, which I will tell you later, that one of my friend is doing that, and he is trying to make hemoglobin in plants, because there is lot of condition of anemia and other thing, and he is trying to make that hemoglobin to express in plant. But I want to come to this topic now. So uh, if the protein is intracellular, the way to break it down is through chemical method, that you need to introduce some kind of a detergent, or you can have a alkali enzyme method. You have to break open the cell. You want, if it's a bacterial cell, you will introduce lysozyme that can break open the cell. But most often the process is either you do it through sonication, which is like you generate sound waves uh, within the sample, and it leads to compression and rarefaction of the sound waves, and because of this high intensity of the sound wave, the bacterial cells are broken down. Other way is to apply a lot of pressure, and this you do through a special instrument, French press. And I had an opportunity to work with both, especially when you want to work with membrane proteins, or the proteins that are attached to membrane. This is the method of choice to break the cell. Mm, this is a little bit harsh, but it works well for globular protein. And so far, I have been able to purify most of the protein which I have worked with till now. We can do simply with sonication and addition of a little bit of chemical method. So once you break the cell, what do you do then is, and this breaking would involve something. So you want to break the cell, but you don't want your protein to be broken down, right? So once you break open the cell, there will be many proteases, and proteases are like enzymes that are cutters of your protein, so they can chop off your protein. So when you do this in vitro experiment, you have to simulate a milieu, and this you do by introducing it to buffers. So buffers are like solutions which can resist changes in pH, and you can choose, depending on the pi of the protein that you are handling, what kind of purification strategy that you want to do. But most of the common buffer is like, what I use is mostly trace, because most of my protein has a pi near neutral pi, and it helps me to select this one. But you can also use HEPs, MOPs, or phosphate buffer depending upon the kind of this kind of the protein it is. And it's not a hard and fast. Here you have to, it's here you have to play a little bit and improvise depending upon the condition and which condition gives you the best result. So you need to have a buffer, of course, and to this buffer you will have to have additives. And the additives are like, within the cell, essentially the environment is reducing, so there is no oxygen within the cell. So to make that, and when you break open the cell, the protein suddenly, uh, and the other component of the cell undergoes oxidation, and you don't want this oxidation to happen to your protein. So then you need to add reducing agent. And the most common is either you add beta mercaptoethanol, but we don't add this beta mercaptoethanol because it has a very strong smell, and people in the lab will get irritated when you use that if you are working with someone. DTT also we don't use, it has also has a very strong smell, but you can use this depending, you can use it in the hood or somewhere where you have proper ventilation. And then you, the what we use in our lab is TSAP, this to read as a reducing agent. You need to add very little, maybe 1 millimolar or something. And then you have protease inhibitors. So this you can buy, and then uh, you need to add, you need metal chelators, because sometime what will happen that there are many metalloproteins which are sitting uh, within the cell, and they are happy there, they don't do much harm. But when they come out, and if there are free metals in the system, they become very very active, and they want to chew your protein and digest them and break them to small uh, components. To inactivate them, you need to add some metal chelators. So these are like um, ethylene diamine tetraacetic acid is one, and EGTA is the one. You can add them, and what it will do is that it will take off and sequester. It will just uh, take all the metal ions that are present, especially the divalent cations, which are important for this enzyme to be active. It will be taken um, from the milieu. So your protein is safe. And then you need to add some kind of osmolyte. And usually we use either sucrose, glucose, or glycerol to stabilize the system. And like most of the protein that we purify, say, with 150 millimolar sodium chloride as the salt. You can use sodium chloride, you can use KCl, or you can use another salt also that enhances the solubility. And this technique is kind of uh, uh, adding salt to get the protein of interest. So one process is called ammonium sulfate precipitation. And the way here is that the technique is based on the solubility of ammonium sulfate salt. So when the salt is not there, the protein is well hydrated. So when you start adding the salt, and it's called ammonium sulfate precipitation cut. So you have different, you add 10%, 20%, 30% like this, and you go on moving till you get your protein of interest in the hand. So what will happen that when you start adding the salt will break into the ion ammonium and sulfate, and this ammonium and sulfate ion will interact with the water. Eventually, the protein will not have enough hydration, and they will come together, and then they will precipitate. And this phenomena is called the salting out effect. One good example is that one method of salting out is that if you remember, पहले के जमाने में क्या होता था कि मतलब पानी को प्यूरिफाई करने के लिए क्या करते थे कि थोड़ा सा फिटकिरी डाल दिए, तो उसमें जितनी भी गंदगी होती थी वो सेटल डाउन हो जाती थी नीचे. So like using that kind of approach, like a salt to precipitate the impurities, here we are using it to precipitate the protein and get the purified protein in hand. This is kind of a first step. Then I want to introduce it to the field of column chromatography, and which has like revolutionized uh, the way we purify the protein, and also the automation that I talked before, that you have now systems whereby you can just hook up your columns, and these are like pre-packed columns that you can use, or you can also pack your own columns. It's easy. So essentially you can do protein separation based on different properties of the protein. If you are doing it based on the size, so exclusion chromatography is one technique. If it's based on charge, how is the charge distribution in the protein, it's called ion exchange chromatography, and it can be either cation exchange or anion exchange. I don't want to stuff you with lot of like internal details, but I am going just superficially a little bit. Then you can also harness how hydrophobic is your protein on the surface, and this hydrophobicity can be used to purify the protein, and this phenomena is called hydrophobic interaction chromatography. And as one of the students rightly pointed out, that you can use biorecognition, like antigen antibody reaction can be used to purify the protein. And you can also uh, like scientists have found that there are some metal, uh, there is a resin called nickel nta, which has a affinity for histidine residue in the protein, and this is called immobilized metal affinity chromatography, and it's one of the widest used technique to purify protein. That can also be used, that you use a hook kind of a thing that your protein can bind to, and later on when you load the protein, all other things are impurities will pass through the column, but your protein of interest will get hooked to the column. So essentially gel filtration, ion exchange, and affinity. And gel filtration, I would say, is or the gel filtration is also called size exclusion, and is, as the name suggests, you are excluding certain sizes. And it's like a, you can say like it's like a chalni, it's like a sieve, but this sieve is different than the sieve that we use to make the tea, because here the pore size are different. So what will happen, that you have a matrix within the column, and you load your mixture of the protein sample above the column. And what will happen, either you can run it under gravity, or you can run in one of the automated system that I showed you. And what happened, that these, they have a small porous matrix which are uh, which doesn't which are chemically inert, it doesn't changes the property of the protein. What will happen, that bigger size of the protein will move through the interstitial spaces and will come first, and protein which are smaller will enter into these beads, and they will come later from the column. So it's just like putting lot of stuff of different sizes on the top of the column and letting them flow uh, where they separate based on their sizes. And this you can monitor, because I told you that most of the protein will have these uh, residues, tyrosine, tryptophan, and phenylalanine, and you can monitor the absorbance of this uh, at 280 nanometer. And then when the protein will come, you will see a big peak coming, and you can have those fractions collected later on. So this is how you do. The other method is the affinity chromatography, and the way here is that you have your protein of interest, and there are mixture of protein that you load into the column. The protein of interest will bind to the matrix of the column, which has a hook, which has a ligand, say, and then the protein and ligand interact, and rest of the protein will come as impurities. So when you wash, most of the protein which is not hooked to the column will come first, and your protein will elute later. And this profile looks something like this. So you load your protein, and you give a gradient of like here it is shown for his tagged columns, where his tagged proteins, where you have a protein which has 6 histidine tags, either in the N terminal part of the protein or the C terminal part of the protein, and it is hooked to the column. So when you wash, all the impurities are gone, and the peak that is seen here, a big peak that you see. So you load the protein, sample loading, then you wash, all the protein is washed off, and when you elute through a gradient of imidazole, which has a similar structure as histidine. So it's like a competitive binding. You go on adding imidazole in a gradient concentration from 0 to 100%. And what will happen slowly, eventually, is that the protein will get eluted. And here it is shown that at 65% or 325 millimolar of imidazole, the protein is getting eluted. So you collect this peak, and this is your protein of interest. And you can go on verifying this protein and testing this protein further. Then comes ion exchange chromatography, and ion exchange chromatography is also a very unique technique. And I will tell you, I I have been very fond of this technique. Why? Because then you don't need any tag or something to put in front of your protein, and then you don't need to cut that tag. So if you can master ion exchange chromatography, this will help you purify the native protein as it is, like you don't have any tag or you don't need post processing. And I must tell you that then I will come into it eventually, that uh, if you have lot of post processing step involved to come to a single band protein that you want, it will eventually dilute your protein at every step. So the lesser the chromatographic strategy, the more the protein you will get in the end. And this is the method of choice for me. So you can either have a matrix, inert matrix, which has a negative ligand or a positive ligand attached to it, and then you put your protein at a in a buffer which a pH which is either higher or lower to the pi of the protein, one pH higher or one pH lower, and then you let the protein bind to the column. When the protein is bound to the column by the virtue of charge electrostatic interaction, what will happen, that different protein that has different propensity of charges distributed on it will elute differently, and your protein which is bound to the column very strongly will elute at certain point. And this you can do by increasing the concentration of the, there are two methods. You can either change the pH of the solution, or you can elute the protein by increasing the concentration of the salt. So you can go say from 50 millimolar salt to say 1 molar salt, and eventually in the process, if you have automated the process, you can see that there at some point you will see a peak coming in, and this is the protein of your interest. And this is a very 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 good method to have concentrated protein of your choice in your hand, which requires very less processing. So as I told you that the matrix can be of two type. It can be an ion exchange or it can be a cation exchange chromatography, depending upon the type of moiety that is present, um, that is hanging out in the matrix. And uh, here we use Q saccharose. Saccharose is just an inert bit to which quaternary amine is attached as the functional group, and that helps in interacting with the protein. And then we also use FP, it's called saccharose column, and this can be used to, this has this carboxyl moiety attached to it, and this helps like depending upon. And one more thing I want to add here is that depending, it's necessary to look at the pi of the protein before you go for devising the strategy of purifying the protein. And then comes the hydrophobic interaction chromatography, which is based on how many hydrophobic residue here shown in yellow. So you can bind the protein to the column, the hydrophobic interaction column. It's called HIC chromatography. You can bind to the protein to the column in a very high salt environment, and what the salt will do is that um, it will let the protein bind to the column. But eventually when you decrease the salt concentration, the protein will elute. If you have a mixture of protein in your sample, it will elute depending upon the degree of the hydrophobicity in the protein. So higher the hydrophobicity, later it will be eluted from the column. And this is how you separate a mixture of protein that is there. So coming back to the strategy, when you purify the protein, there are three steps. One is that you need to capture the protein, and capturing is what I told you, that you need to break open the cell and you need to get hold of the protein. And then there are steps of intermediate purification. uh, so in capture, you isolate the protein, you concentrate and stabilize, and you remove the bulk impurities. In intermediate purification, you remove rest of the impurities, and when you polish it, the protein, you get a single band purified protein. And depending upon your need and the degree of purity that you want in your protein, there are a lot of steps involved here. So if you see this scheme of purification, so first comes preparation, extraction, and clarification, which I told you that you do it through sonication, and you grow the protein express, you lyse the cell, and you remove the cell debris. And you have the supernatant, a solution where you have your protein uh, with other protein. Then to capture the protein, one way is to, depending upon the condition, you might get a fairly pure protein at this stage itself. You capture the protein, and you can do it through ammonium sulfate precipitation, or you can run, um, you can concentrate and stabilize the protein at this stage. Then you go for liquid chromatography, which I told you, either you go for an ion exchange or you go for a IMAC, depending, or you go for a mixture of both. Like one protein I have here, which are working right now, is a protein that is involved in cancer. It's called MEMO1, mediator of cell mobility 1. And here I use two steps. I use ion exchange chromatography coupled with immobilized metal affinity chromatography to purify this protein. And after this purification, you need to polish to get the highest level of purity. And it all depend upon the speed, capacity, and recovery, and how much, like what kind of the quality of the protein you need. So capture, I have already explained that you rapidly isolate the protein. I'm not going to go much into detail, but you use ion exchange or hydrophobic interaction chromatography. You can use also IMAC at this stage to capture. Then you go for intermediate purification, where you remove all the major impurities. And I think for me, till now, ion exchange has been the method of choice here, because you get a fairly good amount of protein in a concentrated form using ion exchange chromatography. Or if you have a tag, you can also use uh, metal affinity or affinity chromatography at this stage. And finally, in the polishing stage, you need to uh, have uh, to remove other proteins which are either oligomers or higher uh, slight impurities which you don't want to be there in the protein. So here it can be all the technique. It can be either reverse phase, you can use gel filtration. For me, so far, because we are not producing antibodies or enzymes that need to be used for therapeutic purposes, we do mostly basic research. And for me, for all the protein that I purify, the final step of polishing is gel filtration. So if you see this, here is the marker, and maybe this is the protein of interest that you want, and you go through different stage of purification, and ultimately you want this protein. Here also, if you see, there are slight impurities, but depending upon the need and what you are using this protein for in your downstream experiment, it will depend. The process will depend. So how do you do this? You just take your sample and you do a SDS page analysis called denaturing polyacrylamide gel electrophoresis, whereby you load your sample, and depending upon the size, uh, it will migrate, and you can have your fairly purified protein at the end of the day, which you can use for your experiment. So we have been talking a lot, and I have shown you the way from one end of the spectrum where you can start with either, depending upon what is the end of use of the protein. You can start either from bacterial cell or the yeast or the mammalian cell, or you can express the protein in the plant. Uh, and one of the model plant-based plant which is used is Arabidopsis thaliana, and also the tobacco plant. Uh, they are well characterized. So, as I told you, that one of my friend who works in Sweden is working on plant-based uh, expression of protein, and he purifies. Right now, his project is to make hemoglobin in plant. So imagine that when you purify the protein, you can see it through your eyes, because hemoglobin has a heme moiety, and that makes the protein red. So you can see them nicely coming through the column. I should have brought some picture, but I don't have it right now. But you can very well um, purify that protein very easily, and you know where the protein is coming. So why there is a lot of interest? I told you that there is a industrial uh, side to it also, that you can make protein, you can sell it in the form of reagents, in the form of chemical therapeutic, in the form of food additives. And one way to do and it has been done for long, that you might have come across this plant algal protein spirulina. So you can grow them in large vats and places. And I, like 20 years back, I wrote an article on spirulina, and there was, like, for vegetarian and vegan people who don't want to have animal protein, that can be a method of choice, that you have protein that is expressed in plant. Or if you are not very eager with the genetically modified proteins to take, you can always have this algal protein. And alga has been used of late mm, in many kind of biotechnological products, and also food, feed, and fuel. And I will give you two example of startups from our own university, which is based on this concept of purifying protein. One is a company called Mycorrhiza. So what essentially Mycorrhiza does is that they have come up with a alternative of meat. People who don't want to consume meat, they are growing algae, which is rich in protein. And a large section of the society which is vegan or vegetarian, and protein is costly, by the way. You like इफ यू बाई इन सिंपल है, आप मार्केट में जाइए, दाल का प्राइस आप देखिए, प्रोटीन का, और जो वेजिटेरियन के लिए दाल दाल ही एक मेन मुख्य सोर्स है. तो वैसे में इफ यू सी दोज प्रोटीन्स which comes in the form of lentil, they are costly. So Mycorrhiza is a startup, and they have a proof of principle, and they are now at a pilot plant stage, where they are making uh, uh, they are growing protein in bulk, and they are not using any genetically modified organism. They are using uh, strains of algae which are already there in nature. So it's completely natural, and they are trying to make uh, patties, protein patties, uh, like burgers. And these one can have as a additive to meat, or it can also be it can stand alone on its own. So and this company is started by an Indian, actually, here. He is a PhD from just a floor above where I work, uh, and he started with this idea, and it's an offshoot of his research project. Another company which is there mostly, which is working here, is Biopetrolia, and this Biopetrolia is also a startup and a company which works mostly with synthetic biology and genetic engineering, and they are trying to use yeast and mm, um, yeast as a source of fuel. And they are making a lot of product. If you go to their website and see, they're trying to really do a lot of things, uh, uh, coming up with new ideas and new biotechnological products. So these two company I know, uh, which are there. But in India, if you go back, there is a Murgabba Chetiyar group which produces spirulina. There's Dr. Reddy's lab, Bharat Biotech, and there are others. I remember that we used to work with Bharat Biotech when I was a PhD student. We used to analyze their samples because we had a facility uh, in CDRI to do that. So there are also many startups and new companies in India which are focused on it, and maybe you will get to know more in these series of talks that you are exposed to now. So with this, I would like to stop and end my talk. But before I end, I'd like to tell you something about whom I work here. I work with Pranila Vitung Staffshed, who is a professor in chemical biology, and we work in a department of biology and biological engineering. The university is Chalmers University, of course, and we are located in Gothenburg, Sweden. And also, I owe a lot of things. I have learned a lot from Pranila, and the other guy who I want to acknowledge is Professor Anil Gupta, who invited me to give this talk after knowing my specialty. And he has been my guidepost from the beginning. Actually, when I was a college student and when National Innovation Foundation was established, and I submitted in that competition, I submitted one entry too, and that's how I came in touch with Professor Anil Gupta. So I'll stop here the screen sharing, and uh, thank you for listening patiently. And now you can ask any question if you have. Yes, thank you.
Hello, sir.
Yes,
Good afternoon, sir.
good
Sir,
afternoon.
I want to know about the additive. Sir, can you show the slide, sir, where the mentions sir ammonium precipitate in solvent, sir?
Ammonium
Which
precipitation.
yes,
Okay.
sir. Uh,
Cool.
for easy convenience, sir.
Yeah, yeah, I am doing that. Oh, I have to go back, I think. Okay, I'll go back. Can you see this slide?
Yes, yes,
Uh, okay, I will go to the. Yes.
yes, sir.
Hmm.
So, sir, here I have confusion. Ammonium
Yeah.
sulfate precipitate. Sir,
Yeah.
actually, what happened in this sir, solvent, sir? Sir,
Yeah.
here ammonium sulfate should be precipitate or not, sir? I mean, sir, if the ammonium sulfate precipitate, then,
Yeah.
sir, any method for the avoid this? I mean, ammonium sulfate precipitate. I'm asking.
Yes. Actually, your protein will be precipitated
Okay,
with the salt. So you can take this protein out, and there will be ammonium sulfate into it, of course, and you dialyze this. Like uh, through a process of dialysis, you can remove this extra salt, and you will have your protein of interest in hand.
okay, okay, sir. Thank. And
So
sir,
this is a very well known established phenomena. If you go and study a little bit about salting out of the protein, you will get this in the textbooks. Yes,
okay, sir. And sir,
This
any
phenomena is called salting out. Yes.
okay, and sir, any other sir application for the purity of the protein, sir? Application.
Application, as I told you, see, there are a lot of industry, like protein is everywhere. You
Okay,
cannot, and like one of the Nobel laureates said at one point of time that life is all about a balance between protein synthesis and protein degradation that happens within the cell. तो हम जीवित ही हैं क्योंकि हमारे प्रोटीन सही काम कर रहे हैं. अगर प्रोटीन सही नहीं काम करेंगे तो क्या होगा? प्लाग्स होगा. If you have plaques, or if you have some enzyme which is dysfunctional, if you don't have protein, you can have protein energy malnutrition. So it's important. There are several aspects to it. Abhi jaise, if you see diagnosis, the entire area of diagnosis, whether it आप इंसुलिन इंजेक्शन लेते हैं, ठीक है, इंसुलिन के मॉनिटरिंग के लिए जो डायग्नोस्टिक टूल है, या जो फर्टिलिटी टेस्ट के लिए, everything is based on antigen antibody reaction or enzyme based assays. So ये कैसे आता है? तो ये it has to be produced industrially.
Okay, okay, sir.
You have papain, you have other enzymes that you need in bulk. So उसको, so there is a lot of innovation that is needed to produce protein. And as I told you, that there is a wider population in world which is malnourished. So protein based food is a very big area to be explored. Also, and an alternative to conventional fossil fuels. If protein engineering can come, or synthetic biology can come at rescue, and you can have a better choice of energy source. So these things are there. Yes.
Or sir, also sir, it is applied on the sir Parkinson disease, sir. No, right, sir?
पार्किंसन में देखिए, हमारा जो अभी द करेंट रिसर्च दैट वी डू
Yes.
इज बेस्ड ऑन, लाइक पार्किंसन पे काम करते, तो अभी तो जैसे रिसर्च इज अ थिंग दैट you don't come with a solution very quickly, right? It takes time. So now we are just trying to understand this protein, and this protein is an intrinsically disordered protein. Intrinsically disordered protein का मतलब है कि इसका अपना कोई शेप नहीं है, it doesn't have a shape, but it requires certain shape when it's triggered. तो हम लोग क्या करते हैं, यहाँ पे पढ़ते हैं कि ठीक है, कोई मेटल आयन अगर हम मेल्यू में डालते हैं, या कोई नेचुरल कंपाउंड डालते हैं, या कोई अदर मॉलिक्यूल डालते हैं, डज इट अफेक्ट द प्रोपेंसिटी ऑफ दिस प्रोटीन टू फॉर्म एमाइलॉइड? उसको कम करता है क्या, या तेज करता है क्या? बट वी डू दिस इन विट्रो स्टडीज, सो देर आर लॉट ऑफ स्टेप्स दैट विल बी इन्वॉल्व इन डूइंग सो.
Okay.
यस.
Thank you, sir.
Sir,
Well, sir, I
sir,
have a question.
Yeah, please go ahead, one by one I can take. Yes, there's a lot of,
Yes, sir.
yeah.
Like, why positive phi angles are less common than the negative phi angles in like protein which contain that L amino acids?
I cannot answer this question immediately because this is very basic question that you have asked, but I can look through and get back to you. But if
Sir,
you know, you can just share. Yes.
no, no, sir. I don't have in the answer basically.
Yeah.
But I have another question. Like that, can the folding of protein be an example of self assembly?
Yes. If you have a small protein, say, it can like my boss, she initially like lysozyme. Lysozyme can self assemble very quickly, and it forms crystal very quickly. That if the protein is very small, it will find a way to assemble itself into its natural shape. And uh, I don't remember this, but I think my guide Pranila, she has a world record of having the smallest protein folding on its own, uh, like this self-assembly thing. I don't remember exactly, but she has one. Yes. Uh,
Okay. Thank you, sir.
sir, in the beginning, you told us that that Europe is one of your mentors told you that the Europe is a better option than US.
yes, I can
So can you
reflect a little bit.
please,
Yes.
yeah, sir.
The thing is that I mostly, if you aspire to be a scientist, people will tell you that you go to US, right? After your training in India. But my boss had a different take on it. He wanted to inculcate the spirit of working together. Like if you go to US, there is a more of an individual excellence. Okay, but when you come to Europe, and especially if you come to Scandinavia, there is a concept called lagom, and lagom is a Swedish word which means just enough. So you they want to co-evolve, and they want to co-create success, so you get due credit of what you do. uh, so that's why probably what I think his idea of um, recommending us to go to Europe was to help us learn more about working in cooperation, uh, and giving our personal ego aside. So working in the greatest common good of science, that you can learn from Europe. And the other thing is that they are very punctual. They are very things happen on time. So that's why I think he might have recommended. That's my idea, because he's no more. I cannot go and ask him, why did you recommend? So eventually I ended up, I haven't been to, I got an opportunity to be in US, but I have chosen to be in Europe. Yes.
So all over Europe, or it's just some
I think it's
certain
more or less
parts?
all over Europe. And if you choose to do a, like, if you do a PhD in, say, Scandinavia, and especially in Sweden, it, PhD is kind of thought to be, you are more or less like an employee of the university, and you are thought to be being trained. It's not a training in that sense. It's being trained as an independent thinker. So at the end of the PhD, you should evolve as a uh, not as a degree holder, just a degree holder, but as an independent thinker and scientist. That's the thing. So there is a kind of a difference between being a guide and being a mentor. So here the philosophy works more about mentoring the student. So it's not about that you finish your course and you are left about to struggle on your own, but you will be nurtured in a way that you will need no more support. I would say you will be independent enough to handle project independently. Yes. So that's the difference.
Thank you very much, sir.
Thank you. Yeah, any more question?
Yes, sir. Good evening, sir.
Good evening.
Sir, I
Yes,
want to
Sachin.
ask one thing, that how we can enhance the thermostability of any protein molecule.
Yeah, this is very good question, I would say, and we do. I have worked with some of this.
Because sir,
Research.
majorly most of the protein molecules are only stable up to 40 or 45 degrees centigrade.
Yes. So
And
ideally, what one way to look at it is that you go for a bioinformatic search of the protein of the similar kind in other organism, maybe in extremophiles, and try to study their protein. The other way around is to go for protein engineering. So you look for residues that is not part of the active site, but that lies in the vicinity of the active site, which is not involved in catalytic activity of the protein. But if you change one or two residue, it can enhance the stability of the protein. And that we have done for some protein, yes. And this is the thing that goes into the industry. So when say you have an enzyme that is needed,
yes, sir.
they want to enhance the stability, and they do it in this way. By protein engineering, one is one way to do this thing. So
Okay, thank you, sir.
changing residues which are not part of the active site, yes, of the protein,
Okay, sir.
and it doesn't affect the folding of the protein in that way, but it changes the thermal profile. Yes.
So is there any rule to it that certain residues can be changed with certain residues only, particular residues?
Uh, I think except proline, which is a chain breaker, you can use other residues, and you have to be careful that it doesn't affect the enzymatic activity of the protein. It, if it enhances, it's good, but it should not deplete the activity, or it should not completely inhibit the activity. So in the area of protein research, it's the folding is very primordial. You have to keep in mind that for a protein to be functional, it should be folded properly, and that's why you have to take care of this. Yes.
So CRISPR-Cas9 is the protein is a technique that we are using as of now
Yeah, CRISPR-Cas
for
is
that.
a very good technique, and it is not only used there, but it can be used at in different arena of science. We are using here CRISPR-Cas, not me, but my colleague is using to silence genes and study important genes which is involved in cancer.
Okay, sir, thank you.
Thank
So I have a question.
yes, Hari.
Um, so in protein engineering, when we use bacterial systems for expression,
Yes.
uh, So these proteins which they produce, be it
Yeah,
enzymes,
yeah,
they have a different post-translational modification than
exactly,
eukaryotes.
exactly. Yes,
For example, insulin, human insulin, which is produced in genetically modified E. coli,
yeah.
it is not. It cannot be directly administered in humans, right? So how do you
Yeah.
overcome that?
So I particularly haven't worked with any therapeutic protein, but I think there are ways in which uh, one has to see whether these are these are important for the functional attribute of insulin or not. If the functional attribute is not hampered by post translation, it can be used by uh, doing a post processing whereby you just remove the toxicity that can come from growing these protein in the bacterial system, and you can use it. But yes, I totally agree, but therefore protein therapeutic is a costly process. Then you need to move from the system. You have to go to either a yeast based system or a insect based system or a mammalian cell culture system. Mostly the vaccines that we have, they are produced in mammalian cell culture system, because there you cannot afford to uh, have something which doesn't which is not functional properly. So that can be the reason. But insulin, I think, is generated through recombinant means, and it has been a method of choice, because I think maybe those post translational modification doesn't make so much difference.
Okay, thank you. Sir, in the previous question, you said something about prolin being a chain breaker. What is that about, sir?
uh, like if the protein has structures, right? So it can either form alpha helix or beta pleated sheets, or it can go into random coil. So whenever there is a proline, uh, it hinders the movement of the and the arrangement of the molecule. That's why it's it's also pedagogical. I learned it long back that proline, if you have proline in the system, it acts as a chain, because they have very specific role to play.
Thank you, sir.
You are welcome.
Sir, I read about this MIDI 88 protein,
uh, Pardon, which?
MYD88.
MYD88. Okay.
So, sir, there was a mutation in that on a certain position, a point mutation, which
Yeah.
changed from leucine to proline.
Okay.
In normal conditions, it was leucine, and in the
Yeah.
mutated protein, proline.
Yeah.
So,
Yes.
in that case, we have it's usually in glioblastoma.
Yeah.
So,
So
we, sir, एक चीज I think that we saw that in the doc when we docked the mutated protein, it
yes.
was it showed more negative binding energy than the
Yes.
wild type.
Yes.
So, is there any particular reason that we can say that a mechanism that we can say that because of this mutation, we saw that the change we saw?
I think maybe, as you rightly said, and I pointed out, and I cannot go back to my master's days about this amino acid thing, but I can tell you one thing. Since the leucine is mutated to proline,
Yes, sir.
this might be the reason of changing the property of the protein, and because of that, it might lose its activity, or it might become something which is not natural, right? And that's why you have this disease manifestation in glioblastoma, as you said. So this might be the cause that instead of leucine, if you have a proline, it can change the property of the protein drastically,
Okay,
and
sir.
that's why you have a different binding energy in your docking experiment.
So we are trying to also do it in vitro, but
Yes.
after COVID, after the restrictions are lifted, we'll
Yes.
try to get into that.
Yeah, it will be a good idea, and you will, and I think then you will explore more about proline and what proline does when you, and I just triggered you to look more into it. Yes.
Yes, sir. Sure, sir.
Yeah. Any more questions? Oh, somebody has asked for an email ID. Yeah, I can share my email ID. I can share it here. You can write to me at uh, drkumar.ranjit at gmail.com. And you can very well ask any career related questions or anything like coming to Europe for higher studies or any kind of question. I will be happy to answer it.
Sir, I actually have one question, career related,
Yes,
if
yes.
you have time, sir.
If the organizer doesn't have any problem, I can I can take this question. Yes.
So the thing is that we have to do internships. I am a BTech student.
Yeah.
So in third year, we have to do internships.
Yes.
It's a mandatory in our course.
Yeah.
So, sir, I have a standing offer from a Canadian university,
Yes.
but I'm also applying for Europe because of Canada has some restrictions as of now as well for the
Yes.
COVID-19. So I'm
Yes.
applying for the European, mailing to European professors as well.
Yes.
So,
So
so
if you
what
have
is,
interest in protein folding, say,
yes,
or
sir.
if you want to work in our area,
Yes, sir.
you can write to me, write to my boss. I have given the reference. You can find about Pranila Vitum staff ship. You can write her directly. You can write your interest. And I know when I joined here in 2016, I don't know which organization you belong to or which institute, but there was a student from ICER who came. He was a bachelor student and who came for his project here. Yes.
Yes, sir. So, according to you, as a comparison, if we say that which
Yes.
would be a better option to
I
get
cannot
into?
say because it depends individually upon the scientist whom you are applying to or who is there. And I think f- science is such a thing that the language of science is universal. You can go to any lab and it will be all the same. So
Yes,
it's
sir.
just depend upon the kind of mentoring that you will get. Yes, I think Canada is also a better choice.
Thank you, sir.
So, any question now?
I think you can write to me directly if you have some career related questions. It will be I will entertain when I have time. That is good.
Yes, sir. So, guys, if you have any question, you can write it to me, or sir has given his email ID. You can directly contact with proper query and purpose. And sir, it was really wonderful session and very informative. And we always look forward for your support and contribution in such future workshop. And
Thank
thank
you.
you so much. I'll
I,
get
I hope that next time when we have a workshop on this, I could give you a lab tour and take you to the real world of protein purification, showing you steps. But
that would be great, sir. Great.
this wasn't possible here because I am not very fan of teaching something practical in a theoretical format.
Right, right, right. Even for us, it is very difficult to organize such virtual workshop
Yes,
because we are researcher and it's
yeah,
it seems like more of academic.
yes, yes.
Yeah.
So.
Anyway,
Uh,
but sir, it was really nice, and I will
Thank you
discuss
for
with
giving me this opportunity to be there and interact with some young guys. Usually, we are lost in a world where I don't teach that much, so I don't really get to interact with his students that much. But
yeah,
it was a welcome change. We have like a one program where I teach, but mostly I am out of this teaching business. But this was a welcome change for me too.
yeah,
Yes,
yeah. Pleasure, sir. Pleasure.
thank you.
Uh,
Excuse me, sir.
Yes,
yes.
Sir, I have a doubt in this protein engineering, sir.
yes. Tell
Sir,
me.
can we apply protein engineering for overcome the Alzheimer's disease, sir?
See, like whatever you do in vitro, right? You cannot go inside. The brain is the most difficult organ to be touched because any where you will find the subject to test your experiment
So
if you do
that's
this.
difficult, sir. But we can modify the protein to it's a type of misfolded proteins which causes the Alzheimer's disease,
Yes,
sir.
yes.
So we can overcome that misfolding of the protein. By
Yes.
that we can.
I will let you know that there are scientists in our research group who are focused in Alzheimer's, and they have been working from last time immemorial, I would say. They are trying to understand the basic property of this. There is a peptide called A beta 42.
Yes,
and
sir.
this peptide is like it's thought to be the culprit. And first of all, to work with this peptide is a real challenge, because the moment you purify this peptide, it tend to aggregate. How
Oh.
will how will you work with such kind of peptide? So I think there is a lot of insight needed, but it's a good idea. It's a good idea. It's a good idea to park it at this point of time and read a little bit more on Alzheimer's. And uh, you can definitely find ways. No idea is a bad idea, first of all. So you read a little bit about it, and I think that's why there is a difficulty in finding cures for neurodegenerative disorders. The
Okay, sir.
maximum you can do is to slow down the process. That's it.
Okay, sir. Thank you, sir.
Yeah, you are welcome.